Welcome to Zoology class. So in this class, we are beginning a new chapter, that is molecular basis of inheritance. So in one of the chapters, we had already seen the principles of inheritance and variation. Principles of heredity or inheritance and variation. Now in this chapter, we will be discussing about the molecular basis for inheritance. What are the molecules responsible for heredity? Or which is the molecule that is responsible for transferring the characters from one generation to another generation? The molecule or the material that is responsible for the transfer of characters from one generation to another generation is known as the genetic material. The material that is responsible for, or the substance that is responsible for the transfer of characters from one generation to another is called the genetic material. So Gregor Mendel, when he proposed factors as carriers of heredity, he had no idea what this could be, what this material or what this substance could be. So he simply proposed factors. Then the study of Sutton and Bovary revealed that the things which Gregor Mendel called factors were located on chromosomes. So he proposed the chromosome theory of inheritance. Then the work of T.H. Morgan and his student Sturtevant revealed further information regarding the genetic material. And they suggested that the factors or the genetic material or the genes were located on the chromosomes and they were arranged in a linear fashion. That means several factors or several genes could be seen on a single chromosome. Even then, it was not clear to scientists what actually this material is. What actually the material is. So the investigation, the research went on in search of the genetic material. And in 1928, Frederick Griffith, a scientist called Frederick Griffith, conducted a very fascinating and very interesting experiment on mice using a bacterium called Streptococcus pneumoniae. You know what are Streptococcus pneumoniae? These are bacteria which cause pneumonia in vertebrates, including human beings. Streptococcus pneumoniae. So when cultured this Streptococcus pneumoniae in his laboratory, he understood that there are two strains of this bacteria. This bacteria exists in two strains. One is smooth strain and the other one is rough strain. So there are two strains of Streptococcus pneumonia. The smooth strain and the rough strain. The smooth strain is so called because the cell wall has a coat of polysaccharide which gives the appearance of a smooth texture. So the cells appear with a smooth surface. due to the presence of this polysaccharide cot. That's why they are called smooth strain. The other strain are rough strain. Because of the absence of this polysaccharide caught over the cell, they appear rough in nature. So they are called rough strain. So there are two strains of bacteria, Streptococcus pneumonia, the smooth strain and the rough strain. As a first step of his experiment, Frederick Griffith injected the smooth strain bacteria and rough strain bacteria into separate mice. Those mice which received smooth strain bacteria contracted pneumonia and they died. But those which received the rough strain bacteria did not get pneumonia and they survived. So from this, one thing is very clear, that the smooth strain alone can cause the disease pneumonia, whereas the rough strain are unable to cause pneumonia. As smooth strain caused pneumonia, they were called the virulent type. Virulent type. Virulency is a term which denotes a pathogenic nature. That means when a bacteria causes some disease, that is called virulent type. When bacteria does not cause any disease, when it is not acting as a pathogen, if it is not acting as a pathogen, it is denoted as non-virulent. So smooth strain is virulent, which causes the pneumonia, and the rough strain is non-virulent, which does not cause pneumonia. Okay, so it was made very clear that smooth strain are virulent type and rough strain are non-virulent type. Okay. Now, in the next step of his experiment, Griffith heat killed the smooth strain bacteria. He killed the virulent type of bacteria, subjecting them to high temperature or heat. So the bacteria died. And this dead bacteria were injected into the body of the mice. Naturally, it is very clear what would be the outcome. The mice will not get pneumonia because the injected bacteria were dead. If they were alive, they would have definitely caused pneumonia. But they were injected into the mice after killing them by subjecting them to heat. So the heat killed virulent, smooth Streptococcus pneumonia were injected into the body of the mice just to make sure that when they are killed, they cannot cause pneumonia. In the third step of his experiment, he mixed the heat-killed smooth strain and the live R strain and injected into the body of the mice. So once again, let us see what he had done in the third step. Mixed a mixture of heat-killed smooth strain, of course, which cannot cause the disease because it is killed. Even though it is virulent, even though it is disease-causing strain, as it was killed, it cannot cause disease. So a mixture of this heat-killed smooth strain bacteria and the live R strain. R strain bacteria anyway would not cause the disease because they are non-virulent. So a mixture of this. So practically both of these bacteria cannot cause disease. Virulent smooth strain is heat killed, it cannot cause disease. Live R strain, it cannot also cause disease because it is non-virulent. And this mixture of heat killed smooth strain and live R strain was injected into the body of the mice. But interestingly, mice got disease. Mice contracted the disease pneumonia and they died. It was astonishing. And why it happened? It should not happen actually. Then how did the mice get disease? From where it got disease? So Griffith suggested that there was some material. There is a material in the smooth strain bacteria that is responsible for the virulency of the smooth strain, or that is responsible for the character of the smooth strain, that he called the genetic material, whatever it may be. And he suggested that this genetic material responsible for the virulency of the smooth strain got transferred into the cell of the rough strain and transformed the rough strain into smooth strain. This is called bacterial transformation. Bacterial transformation. That means one type of bacterium is transformed to another type of bacterium. How? Because here the genetic material responsible for the virulency of the smooth strain was somehow transferred to the cells of the rough strain. And due to the presence of this transferred genetic material from the virulent smooth strain, rough strain was transformed to the virulent form. It was live. Rough strain was live. And it became the virulent type in the presence of that genetic material received from the cell of the smooth strain. So we should understand that even though the smooth strain was dead, heat killed, the heat did not do any damage to the genetic material. The genetic material remained intact even when subjected to heat. So that genetic material got transferred into the cell of the non-virulent bacteria and transformed or converted the rough non-virulent strain to the virulent type. And when it became the virulent type, it caused the pneumonia and the mice died of pneumonia. This was his explanation. So it was made clear by Griffith that there is a genetic material that can be transferred from one generation to another generation or from one cell to another cell. And this genetic material can transform, can bring changes in the cell, can bring changes in the property of the cells. Okay, so this is called bacterial transformation. But even after this experiment, Griffith could not prove what was actually this genetic material. By then, the structure of the chromosome was more clear to scientists. They could understand that chromosome was made up of protein and DNA, a structure called DNA. And they were aware of the presence of another molecule, RNA, also. So this knowledge was there by then. Chromosome was made up of protein and DNA. And the presence of RNA also could be identified in the cells. So they were not sure which is the genetic material. Whether it is the DNA is the genetic material or the protein is the genetic material, or even it could be RNA the genetic material. They were not sure about it. Remember called the structures or factors responsible for characters exist in pairs. Sutton and Bovary told factors existed on the chromosomes. But on chromosomes, in which form? Which is the genetic material? Whether it is the protein or the DNA. So even after the experiment of Griffith also it was not sure, it was not revealed the exact molecular nature of the genetic material. Then three scientists, after the experiment of Frederick Griffith, three scientists, Oswald Avery, Colin MacLeod and McCarthy. Three scientists, Oswald Avery, Colin MacLeod and McCarthy. They conducted experiments. They extended the experiment of Frederick Griffith. They modified the experiment conducted by Griffith to ascertain what is the genetic material, whether it's the protein, DNA or RNA. So they conducted experiments between 1933 and 1944. They followed the same procedure which the Griffith followed. But they modified their experiment. The procedure was modified slightly. They introduced certain enzymes. So when heat killed a smooth strain and live R strain were mixed, they introduced certain enzymes. Say for example, protease. Protease is an enzyme which decomposes the protein and injected into the body of the mice. So why did they introduce the protease enzyme? Because if the genetic material is protein, they did not know what was the genetic material. So if the genetic material is protein, in the presence of protease enzyme, the protein will be destructed. and then it cannot act as the genetic material. So the mice will not get pneumonia. Okay. So from this, they can make sure that the proteins are genetic material. I hope you understand. So protease, so they included the protease enzyme in the mixture of heat-killed smooth strain and live R strain. Assuming that proteins are the genetic material. If protein is the genetic material, this protease would destroy the protein, destruct the protein, hence the protein cannot act as the genetic material, and in such a case, pneumonia will not be contracted to the mice and the mice would be survived. Okay. So when they conducted this experiment, the mice contracted pneumonia and they died. That means, what does it indicate? It indicates that proteins are not the genetic material. Protein is not the genetic material. Then they used RNase, an enzyme, RNase. What are RNase? RNase is an enzyme that destroys or digests the RNA. So they introduced this RNase into the mixture of heat-killed smooth, virulent strain and live rough strain and injected into the mice. Then also mice contracted pneumonia and died, indicating that RNA is not the genetic material in that case. We know RNA is the genetic material that we would see in later classes. But in the case of Streptococcus pneumoniae, RNA was not the genetic material. So mice, even when RNAs were destructed, the character, the bacteria were transformed because RNA was not the genetic material. And the mice got the pneumonia and they died. Then they introduced DNAs. DNAs are enzymes which destroy or digest the DNA. When DNAs was introduced, it is a mixture of heat-killed, smooth, virulent and rough strain of bacteria and injected into the body of the mice. The mice did not get pneumonia and they survived. What did it indicate? It indicates that DNA is the genetic material. That is why when it was destroyed by DNAs, the bacterial transformation did not occur. Rough strain did not become smooth strain. And the mice did not get pneumonia and they survived. So, by this experiment, it was made very clear by Oswald Avre, Colin MacLeod and McCarthy that DNA is the genetic material. Okay. So, it was made sure that DNA is the genetic material. Okay. We shall end up the class here.